Hello, everyone, and welcome to Workshop 28, Systems for Developing and Testing Therapeutic Strategies. I'm Jennifer Gimbalot from the University of Alabama at Birmingham, and I'd like to introduce my co-chair, Dr. Cal Parekh from the University of Iowa. We'd like to thank all the presenters for their excellent abstracts and for presenting today. We would also love to hear from you, so please submit your questions via the Slido platform. As a reminder, we will stream the pre-recorded talks followed by a few minutes for dedicated questions. When submitting your question, we'd love to know who you are and who your question is directed to. So without further ado, please welcome our first speaker.
Hello. I'd like to begin with thanking Drs. Parekh and Gimbalot for the opportunity to present this project entitled Pharmacologic Response of Rare Cftr Folding Variants is Mediated by a Silent Polymorphism that Alters Ribosome Velocity. Highlights of findings presented are also available at poster 649. My disclosures for this presentation encompass sources of funding, which are shown here. Before presenting the work, I'd like to acknowledge many collaborators who have contributed at various points in this project, specifically highlighting past members of Zoya Ignatova's laboratory at the University of Hamburg. Drs. Rauscher, Bompi, and Kirchner assisted with the majority of findings you will see today. I'd also like to thank Disha Joshi and Dr. Eric Sorscher's laboratory at Emory, who has helped tremendously with recent advancements in the work. Ribosome velocity is a topical yet understudied contributor to Cftr biogenesis. Many aspects of translation related to mRNA decoding, co-translational folding, cellular quality control mechanisms, and among others, significantly impact the speed at which Cftr is synthesized, which in turn influences final protein conformation and function. This presentation will focus on the importance of tRNA availability during Cftr biogenesis. At the codon level, a major determinant of ribosome velocity is the concentration of cognate tRNA. tRNA availability is important in the context of synonymous or silent nucleotide polymorphisms, or SNPs, which, according to NHGRI, are variations in a single base pair where the altered codon corresponds to the same amino acid. This point is illustrated below with threonine. ACU in the transcript is read by an amino-acylated tRNA charged with threonine, which possesses UGA in the anticodon loop. When a silent SNP is introduced such that ACU is mutated to ACG, this codon is now read by tRNAs charged with threonine that harbor the anticodon UGC. Therefore, no change occurs in the amino acid sequence, but there is an exchange of tRNA species pairing to the affected codon. Findings presented today are intended to highlight the potential for synonymous SNPs to significantly impact Cftr folding and sensitivity to pharmacocorrection, which is mediated through tRNA-based interactions. The Ignatova laboratory and others previously described a number of silent Cftr SNPs that naturally occur in the general population. These SNPs are depicted in the Cftr tertiary structure on the left and localized to several different subdomains. The most prevalent synonymous SNP detected among healthy individuals sampled from the 1,000 Genomes database was T2562G, which I'll refer to as the TG SNP from now on. Moreover, this SNP commonly occurs on CF alleles that do not harbor a copy of delta F, as evidenced in CF cohorts from Canada and the US, provided through NCBI's dbGaP database. Plasmids encoding cDNA constructs of the silent SNPs were transiently introduced into parental CFBE and assessed by Western blot for effects on Cftr maturation. Densities of Cftr bands are quantified on the right and normalized to both actin loading control and neophosphotransferase, which is the transfection efficiency control as it is encoded on the same plasmid. Some synonymous SNPs augment expression of the mature band C glycoform, whereas others decrease total production of Cftr band C and or the immature protein band B, the most prominent of which was the TG SNP. Interestingly, however, this SNP did not alter Cftr mRNA levels, suggesting that effects were elicited at the level of translation. We employed ribosome profiling coupled to RNA-seq, also referred to as RiboSeq, to determine whether the TG SNP altered Cftr translation kinetics. Unprotected RNA was digested away, and the resulting ribosome-protected fragments were submitted for sequencing. Accumulated reads of ribosome footprints were then assembled and quantified across transcripts. From RiboSeq data, we measured the ribosomal A site dwelling occupancy of all codons, which is plotted here as codon frequency normalized to the average number of footprints across all genes and compared to genome codon usage. As described earlier, the TG-SNP exchanges the wild-type codon ACT or ACT in the DNA to ACG. Ribosomes decoding ACG are slower than those reading ACT, as evidenced by a higher A site dwelling occupancy above the 90th percentile. In addition, tRNAs pairing to ACG are less abundant than tRNAs coding ACT, suggesting that the silent TG-SNP reduces local translation speed in a tRNA-dependent manner. We next sought to determine whether the TG-SNP exerted effects on mutant CFTR processing. The TG-SNP was introduced in cis with the common Delta F508 variant and transiently transfected into parental Fisher rat thyroid, or FRT cells, followed by Western and short circuit current analysis. While the TG-SNP augmented Delta F band B, shown here and quantified on the right, it did not significantly enhance maturation to band C, as compared to correction with VX809, for example. Boosting chamber studies also revealed that the TG-SNP did not alter Delta F508 ion transport phenotype, either alone or in combination with VX809. Interestingly, this silent SNP modestly augmented Delta F508 band B proteolytic stability in CFBE. Limited trypsin digestion revealed slight stabilization of the immature glycoform, as well as a lower molecular weight band indicated by the arrow. In the two CF cohorts mentioned earlier, the TG-SNP largely segregated with alleles that did not carry the Delta F variant. Therefore, we evaluated whether the TG-SNP exerted cis-acting effects on a subset of rare CFTR mutants represented in the patient population sampled. As before, the TG-SNP was introduced into cDNA plasmids carrying the variants listed here along the CFTR transcript and shown in the tertiary structure, as well as in tabular format. They include G85E, G551D, D579G, D614G, F1074L, and N1303K. Mutated codons observed among individuals with CF are provided, as well as the concentration and fold change of each cognate tRNA, which was quantified by tRNA-based microarrays performed in CFBE. In addition, we engineered silent polymorphisms intended to alter the speed at which CFTR variants are decoded. Disease-associated codons that paired to highly abundant tRNAs were exchanged for synonymous codons that paired to less abundant tRNAs, or vice versa, in order to quantify tRNA-dependent effects on ribosome velocity. For example, the mutated codon that naturally occurs in D579G patients pairs to a tRNA with a concentration 20-fold less than the wild-type tRNA. In contrast, the engineered revergent codon pairs with a tRNA that is only 2-fold lower than wild-type. If participants would like to spend more time reviewing this table, please note that it is published in our paper referenced here. Each of these constructs were initially assessed by Western blot. Wild-type or individual missense variants are shown in black. Each CF-associated variant with the TG-SNP in cis is indicated in medium gray. Individual missense variants engineered as revertants, which still encode the TG-SNP as well, are illustrated in light gray. The first box shows band B levels, and band C is quantified in the second box. Both are normalized to actin and neophosphotransporase as before. I'd like to draw your attention to G85E, D579G, and D614G. The TG-SNP appears to amplify expression of bands B and C for these variants. I would also like to point out that some engineered revertants exhibit notable alterations in CFTR production. For example, band C expression of the G85E revertant is significantly diminished, whereas band B expression of the F1074L revertant is robustly elevated. Using qRT-PCR, we did not observe demonstrable changes in relative mRNA abundance for any variant tested. Transient transfection of these constructs was also performed in polarized FRT to assess impact of the TG-SNP on modulator responsiveness. In the interest of time, I would just like to highlight D579G and D614G variants. Biochemical and functional analyses revealed that the TG-SNP alone augmented production of D579G bands B and C, as well as forskolin stimulated short circuit currents. This silent SNP also increased D579G sensitivity to VX809, as evidenced by greater band C expression and transepithelial ion transport. Moreover, maturation and channel activity of the D579G revertant were further augmented and appeared additive with chronic VX809 treatment. In the context of D614G, the TG SNP alone modestly increased short circuit currents, yet significantly enhanced VX809 dependent protein maturation and ion transport. In addition, the D614G revertant further improved band C formation and channel activity with synergy observed with VX809. To assess translational relevance, we plotted percent rescue of CF-associated variants relative to wild-type levels of short-circuit currents. The total forskolin plus VX770 response is shown in the presence or absence of the TG SNP, and with or without VX809. Engineered revertants are not shown. Notably, the TG SNP enhances D579G activity up to 40% of wild-type, and when combined with VX809, reaches 70% of wild type. In the setting of D614G, the TG SNP augments VX809 stimulated currents from approximately 17% up to 30% of wild type. For G551D, the TG SNP increases corrector-mediated rescue of the CFTR channel up to levels comparable to those of wild type. In contrast, however, the TG SNP does not appear to influence transepithelial ion transport of F1074L, either alone or in combination with VX809. Similar observations were made for G85E and N1303K. Taken together, findings indicate that synonymous SNPs can significantly impact mutant CFTR processing and that tRNA abundance may serve as a larger contributor to CFTR folding trajectory than previously appreciated. To summarize, the TG SNP decreases ribosome velocity by approximately 50% at 3-enene 854 in CFTR, thereby altering folding trajectory. This point is illustrated in the figure showing that the wild-type codon ACT is decoded faster than the mutated codon ACG. In turn, this local reduction in speed modestly diminishes CFTR expression and channel conductance, the latter of which I did not have time to describe, but may be referenced in the manuscript below. It is interesting that this mildly deleterious silent SNP, when paired with a disease-causing variant, can epistatically improve mutant CFTR processing and/or function. The phenomenon was observed primarily for D579G and to a lesser extent D614G, whereas F1074L and N1303K were largely unaffected. In the context of D579G, this SNP amplifies band B and band C expression, augments channel activity, and enhances pharmacologic response. Engineered silent SNPs or revertants also exerted positive or negative effects on CFTR processing, providing additional evidence that CFTR folding and function can be robustly influenced by tRNA abundance. It is important to note that tRNA availability does not alter ribosome velocity and/or protein folding trajectory in isolation. Other contextual features will elicit their impact in tandem, such as the primary effect of any modified amino acids, secondary structures, modifications, pause signals, or other protein binding motifs in the surrounding sequence, mRNA surveillance pathways, and many more. Our findings argue against neutrality of silent polymorphism or silent single nucleotide polymorphisms during protein biogenesis, highlighting ways in which silent mutations can change local kinetics of mRNA translation and epistatically modulate outcomes of CF-causing variants. Such effects are likely to influence the spectrum of disease symptoms, represent a mechanistic contributor to genotype-phenotype relationships, and ultimately may help predict therapeutic response in precision theratyping studies. I'd like to end by announcing that our pediatric research enterprise at Emory and Children's Healthcare of Atlanta is growing. We are recruiting basic clinical and/or translational investigators with an immediate opening for a mucosal immunologist shown here. Furthermore, my own laboratory is hiring students and postdocs. If you are interested in either of these opportunities, please do not hesitate to contact me. Thank you for your attention, and I would be happy to take questions.
And thank you for that excellent talk, Dr. Oliver. So we're moving on to questions. I think that we have quite a few people joined, so please send in your questions. I'd like to start with a question that I have, and that is that the SNP that you're referencing throughout your talk What is its frequency among CF patients rather? Or do you have any information about how prevalent this SNP may be among the population? And would you envision a future where genotyping for silent SNPs may be part of precision medicine for CF?
Those are multiple great questions. So the TG SNP is quite prevalent among the general population. It's so prevalent, I wouldn't even say it's got a minor allele frequency. It's about 50-50 of the T or the G present. So among CF patients, there is a higher prevalence of the T nucleotide. But it was interesting when we stratified between CF patients who either had the T or the G, that there was an enrichment of the G for patients who did not carry the delta F. But otherwise, if you just surveyed the entire CF population, then overall there was less prevalence of the G. And then the other part of your question about if genotyping for silent SNPs could be part of precision medicine moving forward, I think it's entirely possible. There are newborn screening programs, for example, that are now implementing a next generation sequencing approach where when a child has their newborn screening indicative of CF, that their first step is to do full Cftr sequence analysis that includes both the coding and non-coding regions. And then, if CF associated variants are identified in that first pass, then, great. But if they're not, then maybe on the latter end, more robust bioinformatic analyses could be brought in to then assess if there are other meaningful silent polymorphisms or intronic SNPs. So I would say that may be a direction we are moving toward, but not quite there yet.
Great. All right, I have another question for you. Patrick Harrison asks, do you have any silent revertants that rescue F508del? And he notes that he missed the first couple of slides. So if just a FYI.
Okay. So I would say, no, we haven't found any yet that robustly rescue delta F. For this TG SNP, we did find that the proteolytic stability of delta F was enhanced. It's just the overall maturation efficiency and or short circuit current and measurements were not robustly augmented. But we do have additional SNPs that were mentioned in that first slide that Dr. Ignatova's group investigated that we are intending to evaluate in the context of delta F as well as these other rare variants.
Okay, great. Let's see. Another question for you. Have you stratified, or has anyone stratified patient responses among people taking modulators of any kind looking for this SNP?
Not that I'm aware of, but I think that's a great idea.
Okay, we just have another question coming in. Very interesting work. This is from Hillary Valley. Very interesting work. You showed differential effects of this SNP on several CF-causing CFTR mutations. Do we know enough yet to predict why this SNP has positive effects on some mutations and negative effects on others? Do we know if the mechanism of this difference is entirely translation speed?
I would maybe answer that latter question first, which is this entirely dependent on speed? And I would say definitely there are other factors involved. The overall translation kinetics are impacted by, sequence context and the tRNA availability, a lot of other examples that I mentioned in the introduction to the talk. And so now I don't think it's working in isolation by any means. And so the first question about, do we know enough to predict if this SNP has positive effects on mutations and negative effects on others? I would maybe just say limited to this study, we observed that the CFTR variants that were upstream of the TG SNP seem to be at least modestly improved by the TG SNP, but the CFTR variants that were downstream of that SNP were not impacted, so such as the F1074L and then 1303K. So it could be something to do with that, or it could be something to do with during co-translational folding, those altered interdomain contacts. I feel like a lot of additional investigations in that space that are needed to really give a clearer answer.
Great. So as we wait for more questions to come in for the audience, I have another question for you. It looks like we have just a couple minutes left. So if anybody has any questions, go ahead and put them in this, the Q&A for us. But my question for you is really relating to the development of novel therapeutics. So when pharmaceutical companies and academic investigators are developing new compounds or approaches, how might your findings be used to help tailor their development?
Yes, so we found not only in this study, but also in previous work that we did with Zoya Ignatova's group, that depending on the cell model you use, that there are sometimes very different tRNA abundance profiles between them. So, one might speculate that depending on the overall tRNA-OM profile, you could get a different folded CFTR, even if it's wild type, depending on the cell system in which it's synthesized. So that very well may partially explain why in certain cell systems we see modulators giving a robust effect and in others not so much, because there's actually a slightly different folded version of wild type CFTR. So yeah, I think it's very important to just keep the underlying genetics in mind when you're working with these cell systems, and just to use mutually reinforcing models. So, as people tend to do, if they find an intervention of interest, to evaluate it not just in FRT or CFB or primary bronchial or nasal epithelia, but across a wide array of those, and as well as in animal model systems. And I think maybe just as a slight anecdote before our time ends, that I'd like to say that we are really grateful for the investigators at UNC Chapel Hill and University of Washington for depositing the CF cohort data that we use from dbGaP, and we look forward to hopefully seeing Dr. Cutting's new CF genome project results as well in the future, that we might be able to utilize that information and answer more of these questions addressed or proposed today.
That's great. Well, thank you so much for your talk today and for answering all of our questions. We're going to wrap up this Q&A and move on to our next speaker. So thank you all very much. Great job.
Thank you to the organizers and session leaders for giving me the opportunity to present today. My name is Elisa Bell, and I am from the CF Foundation's lab. Today, I will be taking you through the development of two CFTR gene-edited cell lines capable of enabling a new, highly sensitive read-through assay, with the goal of performing high throughput screens to identify compounds to provide therapeutic benefit for people with CF with nonsense mutation. There are no relationships to disclose. Approximately 10% of people with cystic fibrosis have at least one nonsense mutation, also known as premature termination codon or PTC mutations. Identifying and developing effective therapies to treat the basic defect for PTC mutations remains a serious medical need. Our goal was to create a robust cell-based reporter assay that can be used as a tool to identify compounds capable of reading through PTCs. This slide shows our strategy to develop an assay to identify compounds that allow for read-through of a CFTR PTC variant. Here is a representation of normal CFTR mRNA, the black line, and the resulting CFTR protein. We engineered a CFTR PTC mutation indicated by the red stop sign, containing a C-terminal reporter indicated by the green star, to be able to evaluate read-through in a high throughput context. When there is a PTC mutation, the ribosome stops prematurely at the PTC, and a truncated protein is produced, shown in red. The next few schematics show the predicted effects of read-through modulators and Nmd inhibitors alone and in combination in this CFTR PTC C-terminal reporter context. Read-through modulators allow for some PTC transcripts to be read through by the ribosome, creating a full-length protein, which in this case also contains a detectable reporter tag. Since read-through is not very efficient, truncated protein will also be produced, but will not be detected by the reporter. A key feature of this assay is that truncated proteins will not include the reporter. The reporter will only be included if full-length protein with the C-terminal reporter is produced. The Nmd pathway degrades mRNA transcripts containing premature stop codon. Nmd inhibition alone results in increased transcript and resulting protein. However, the protein produced will be truncated, and in this context will not contain the reporter. So the assay should not detect Nmd inhibitors alone. Finally, a combination of read-through modulator and Nmd inhibitor should boost the reporter signal, as the transcripts will both be stabilized by Nmd inhibition and read through at the PTC stop codon with the read-through modulator. In summary, this assay should allow us to specifically detect read-through modulators in a high throughput context because of the reporter. We chose a Nanolube reporter tag from Pomega. It is small, about 19 kilodaltons, and emits luciferase. Our gene editing approach was to insert the Nanolube reporter into the CFTR locus at the C terminus. We started in the background of a human bronchial epithelial cell line, 16-HBE-GE, with an R1162X PTC. We nucleoaffected the cells with CRISPR CPF1 for targeted insertion of Nanolube in frame at the 3' end of the gene, and then sorted for single cells to get clonal cell lines. Here is the desired insertion with CFTR R1162, and then at exon 27, it's followed by a 10 amino acid peptide linker, the Nanolube reporter, and CFTR's native TAG stop codon. All the cell lines I will be talking about are CFF 16-HBE-GE cell lines, based on the parental 16-HBE-14O- cell line from Dieter Grunard's lab. But for simplicity, I will use the abbreviated names underlined here throughout the presentation. On the left here is a diagram of the CFTR protein with Nanolube at the C terminus, also referred to as Cftr Nanolube. We have been evaluating potential read-through and resulting Nanolube signal in two ways. A high-throughput screening-compatible Nanolube assay that utilizes a cell-permeable substrate that will react with CFTR C-terminal Nanolube and allow for quantifiable readout of luminescence or full-length protein. The NanoGlow InGel is a second method that involves visualizing the fusion protein, in our case, CFTR Cftr Nanolube, in a protein gel by imaging the luminescence directly in the gel. We identified several clonal cell lines with Nanolube inserted, and a few of them are shown here in the Nanolube assay. This assay, we used a combination treatment that included SMIG1 inhibitor, an Nmd inhibitor, G418, a read-through modulator, and two vertex correctors, VX445 and VX661. In this talk, I will commonly shorten this to SGVV. On the left side of the graph, we see the parental cell line, R1162X no Nanolube, and you can see that there is low background in the non-Nanolube cell line. On the right side of the graph, we have 5 R1162X Cftr Nanolube clonal cell lines with vehicle in gray and SGVV treatment in the green color. DF9 was selected due to its robust assay window, and I will show its characterization in the next few slides. Next, we evaluated the CFTR Nanolube fusion protein expression. This slide shows results from a NanoGlow InGel assay. The first sample is wild-type CFTR extracellular loop 4, Nanolube, a previously characterized archival sample, not matched for protein loading, but included for an approximation of wild-type CFTR's molecular weight. The next two lanes are R1162X parental no Nanolube, and they do not have Nanolube, so they do not show signal. The R1162X Cftr Nanolube signal shows detectable protein with SGVV treatment, and the protein banding pattern is consistent with mature CFTR, indicating that SGVV treatment allowed for read-through to create a full-length protein. Here's a closer look at the R1162X Cftr Nanolube cell line and how it responds to various treatments in the Nanolube assay. Remember, this assay was designed specifically to detect read-through compounds, but the assay window may be boosted with Nmd and inhibition and modulators. On the right side of the graph is R1162X Cftr Nanolube, and conditions that include G418, the read-through modulator, are highlighted in green. We see a strong response only in combinations that contain G418, with the strongest response being SGVV at almost 5-fold increase over vehicle. Meanwhile, the SMIG1 inhibitor and the vertex compounds by themselves without G418 have minimal effect on the signal. This indicates that it is a sensitive read-through specific assay. We also did sequencing. Whole genome sequencing showed that Nanolube is inserted only once in the genome and it is located in the CFTR locus, indicating the Nanolube signal we see is specifically from CFTR read-through. Sequencing also showed that Nanolube is inserted correctly in frame at the end of exon 27, followed by the TAG stop codon. There is also about a 580 base pair duplication of the 3' UTR. Additional clones were sequenced, and these clones also contained 3' UTR duplications. However, each was different. This indicates that the duplication seen may be a feature of the gene editing strategy used. Importantly, although there is a tandem duplication of the 3' UTR sequence, the DNA sequence is expected to produce the desired CFTR Cftr Nanolube fusion protein, which is what we detect in the nanoglow in gel protein analysis. What we've talked about up until now has been the R1162X Cftr Nanolube reporter, diagrammed here. We wanted to see if we could improve the assay window by inserting the EF1 alpha promoter to boost expression of a PTC-containing gene. Here you see our gene editing approach to insert the EF1 alpha promoter into the 5' region of the CFTR gene in the R1162X Cftr Nanolube cell line. We used CRISPR-Cas9 homology-directed repair to insert the EF1 alpha protein. Here's the desired outcome, with the EF1 alpha promoter driving the expression of CFTR R1162X Cftr Nanolube. We used a similar pipeline to create this reporter cell line and identify clones. After clones were selected, we ran a Nanolube assay, looking for an increase in signal with SGVV treatment. For comparison of scale, I've included the parental R1162X Cftr Nanolube cell line here. Multiple EF1 alpha inserted R1162X Cftr Nanolube clones showed a large assay window in the Nanolube assay for vehicle versus SGVV treatment. Due to its robust assay window, CF10 was selected as a lead candidate for further evaluation. We wanted to see if the increased Nanolube assay window observed corresponded to increased protein expression of the CFTR Nanolube fusion protein. The top panel here is the nanoglow in-gel assay that will detect CFTR Nanolube fusions. The middle panel is a Western blot using the UNC 596 antibody to detect CFTR, and the lower panel is the sodium-potassium ATPase loading control. With SGVV treatment, the EF1 alpha R1162X Cftr Nanolube cell line shows a strong signal in the nanoglow in-gel assay. We were also excited to see that the EF1 alpha promoted C-terminal Nanolube is also detected in the Western blot. Again, the protein pattern is consistent with mature CFTR. In summary, this data shows that insertion of an EF1 alpha promoter boosts the expression of CFTR Nanolube fusion protein. We characterize this EF1 alpha promoter cell line in the same way as its parental cell line. Conditions that contain G418 are highlighted in green. Again, those conditions are the ones that show strong increase in signal, while treatments without G418 show minimal signal. This indicates that it is a sensitive read-through specific assay. We also wanted to see if the EF1 alpha promoter cell line would show good sensitivity for evaluating CFTR in the TEC24 assay. Here are the representative traces for the EF1 alpha R1162X Cftr Nanolube cell line, and the corresponding area under the curve quantification shown below in light blue. Also included in this graph are its two parental cell lines. All three cell lines show little to no activity with vehicle treatment. With G418 treatment, all conditions show an increase, and the parental cell lines show similar CFTR function in response to G418. The addition of the EF1 alpha promoter boosts detection of CFTR function. Taken together, this data shows two key points. The C-terminal Nanolube tag does not seem to adversely affect CFTR function, and the addition of the EF1 alpha promoter shows a readily detectable increase in CFTR chloride transport activity in response to G418 and G418 combinations, while the background vehicle-treated signal remains low. The goal of creating the EF1 alpha R1162X Cftr Nanolube cell line was to be able to use it as a tool for drug discovery. Here is a reminder of the read-through assay strategy. Can this cell line be used to look for novel read-through modulators? Here, our team is evaluating high throughput screening characteristics like assay window and reproducibility. 11 384 well assay plates were tested for vehicle versus G418, and the Z factor score was high. The EF1 R1162X Cftr Nanolube cell line shows potential to enable a robust and sensitive high throughput assay. We are excited to see the results of ongoing studies to use this model to find new read-through compounds. In conclusion, we performed 3 sequential gene edits in 16-HBE 14O- cells to introduce the R1162X PTC mutation, to insert C-terminal nanolube, and to insert the EF1 alpha promoter, yielding the 16-HBE GE EF1 alpha R1162X Cftr Nanolube cell line, which enables a robust, high throughput drug discovery assay. The assay primarily reports on read-through and not on NMD inhibition. The EF1 alpha promoted cell line shows readily detectable increase in the TEC24 assay when treated with G418 compared to its parental cell line. EF1 alpha promoter insertion may be a useful strategy to boost CFTR expression in a variety of assays and model systems. Of note, these cell lines are available with an MTA. I'd like to thank the wonderful team at the CF Foundation's lab. A special thanks to those mentioned on this slide in the electrophysiology, genomics and bioinformatics, and gene editing team. I'd also like to thank leadership at the lab, and the whole lab is just a wonderful place to be. For additional information, please see posters 672 and poster 591. Thank you again.
Thank you so much for that excellent talk. We have some questions rolling in from the audience, and so I'd like to go ahead and get started with those. Patrick Harrison asks, how do correction levels compare to wild-type CFTR C-terminal nanolube?
Hi, thank you. That is an interesting question. So for this particular project, we wanted to make a high throughput screen. So we started in the R1162X PTC background. We have previously made ECL4 nanolube that we have wild type. We do not have wild type CFTR C-terminal nanolube. We just jumped right into the R1162X background. So I do not know sort of the correction levels with respect to the wild type CFTR C-terminal nanolube.
Okay. I have a question for you. Are there differences in read-through and nonsense mediated decay across tissue types? And have you planned to use this approach in any other cell types, like from the gut or sweat gland, to see if, like, to add to the novel compound screening?
Yes. So, I mean, I think there definitely it would be reason to believe there are differences in read-through and NMD in different cell types based on sort of what is in the different cell types, what genes are sort of active and pipelines are in those cells. For us, the goal was essentially to create a read-through specific assay. So we wanted something that's going to see read-through compounds as opposed to NMD inhibitors. So with this, our plan is to use this, and it's currently being used at the lab in high throughput screening to identify compounds. The next step, once compounds are identified, would be to see how they respond in 16-HBE R1162X without a tag. We assume they'll respond similarly. And then finally, it would be to use them in primary HBEs to look at current. So I think we'd get a sense of sort of different cell types based on the compounds that come out of it, and then testing it in other assays, other model systems as well.
Okay, thank you. All right, we have a question from Rihanna Lee. I think you may have addressed this a little bit just now, but maybe you can expound upon it. Have you tried comparing your model to primary human bronchoepithelial cells? And what level of correction in your model would be needed to see functional correction in such cells?
Yes, that's a great question. And this model is new, and it's in the sort of high throughput screening phase right now to look for compounds. And then, as I said, the next step would be to take those compounds into primary HBEs and look at what we get in that. yeah, I'm not sure sort of what level of correction we would need to see to sort of compare to the functional level, but that's something that hopefully we'll have some sense of in the near future.
Okay, great. All right, I have another question for you. So I'm encouraging the audience to put questions in as we approach the end of our Q&A for this. But while we wait for that, one of the things that I'm particularly interested in is understanding the differences in how different people express drug metabolism and transport proteins. And so that is something that does happen in airway epithelial cells. And since at this time, you're focusing on the 16-HBE as you introduce this really great technology, have you given any thought to whether differences in the expression of metabolism proteins or transport proteins might impact your ability to screen these compounds in that particular cell line?
Yes, I think that's a great question. It's definitely something to keep in mind with all of these sort of model systems, is that you may see something in one model system and you need to sort of look at it in other model systems and make sure that you see a similar response. So yeah, I definitely think that's something worth looking into, but we decided to focus on the 16 HBEs to get us started.
Okay, great. So I have another question from Carlos Perez Garcia. This is also very similar, which I think we might have addressed, but just to go back, he says, a great assay. The data was generated in 16 HBEs. Will you expect to see a similar efficient response in human bronchoepithelial ALI? So you've kind of addressed this, but I'm not sure specifically about ALI, if that might be a difference
Yes, so we did run these in the 16 HBEs. We ran them in a Tech24 assay and saw a response there. And the plan would be to take the compounds that we get out of a high throughput screen and put them into primary HBEs on ALI, and also run them in electrophysiology assays. And, I think once we get a chance to do that and compare, it will give us a sense of sort of how predictive this model is. And we're hopeful that it will be predictive.
Okay. So just checking to see if we have any more coming in from the audience. And if any of the other speakers today, please, I think we can also chime in.
I would like to just say that if anybody's interested in these cell lines, they are available through an MTA. So please, I'm sure my email is associated with this talk or with the poster 672. Please reach out to me and we can discuss that.
That's great. It's a great resource and really just an amazing technology. So I'm really hopeful to see where this goes next. I think, oh, here we go. Here's one. So from Tyler Brown, do you have a sense of how many compounds could be read in a single assay, and how fast is the throughput?
So we do have the capability to do high throughput screens, and we do these screens by putting these cell lines into a 384 well plate, and we run multiple plates at a time. So it's definitely a high throughput screening compatible assay. So it's
great. I think we have one more question that we can get in the last minute that we have. This is also from Rihanna Lee, who has a technical question. How easy are these modifications to make? Would it be feasible to make similar cell lines with other PTC mutations and also beautiful talk?
Thank you. So this is a gene editing pipeline that we routinely use for the 16 HBEs. It takes several months to go from start to finish. It involves sort of nucleofection, single cell sorting, identifying clones, then characterization. It certainly can be done. Right now, sort of with the R1162X, we started there, and the plan would be to screen for the compounds in R1162X and then sort of branch out into other PTC mutations sort of in downstream assays as well. But it's certainly feasible to make other cell lines that include different PTCs.
All right, well, thank you so much for an excellent talk today. I think we'll wrap up. And if we have additional questions that come in, we'll address those at the end. Thank you so much.
Welcome all. My name is Marlou Bierlaag. I'm a second-year PhD student at the University of Utrecht in the group of Kors van de Ent and Jeffrey Beekman. And on behalf of all the authors listed here on this slide, I'm very excited and happy to share with you the results of our abstract called HitCF Organoid Study ELX02 on intestinal organoids from people with CF with ultra to ultra-rare nonsense mutations. Not much to disclose other than that this project has received funding from the European Union's Horizon 2020 Research and Innovation Program. And in the upcoming 15 minutes, I would like to tell you a bit about the HitCF Europe project, on how we use the intestinal organoid model, a bit of background on the investigated medicinal product ELX02, of course, the results of the organoid screen, and share some future plans we have in the project. HitCF Europe started in 2018 with the main goal to get drugs available for people with CF with ultra-rare genotypes. So the group that currently falls out of the scope of the current commercially available modulator therapy, either because their genotype doesn't fit or that they just simply don't respond to the drugs. As an approach, we use the intestinal organoid model as a predictive tool for clinical drug response. With the help of the ECFS clinical trial network, we were able to locate the patients with rare mutations throughout Europe, approach them to take a rectal biopsy, culture those into intestinal organoids, and use them to test drugs on so that we can eventually invite them to participate in clinical trials. The project is carried out under the HitCF consortium, which consists of 2 CF organizations, ECFS Clinical Trial Network and CF Europe. 4 different laboratories are connected in Utrecht, Leuven, and Lisbon. We have 2 pharma companies aligned and 2 supportive companies. And a little bit about the intestinal organoid model for those who are not familiar with the intestinal organoid models. It all starts with taking a rectal biopsy, which is a very easy and pain-free procedure and can be performed in the outpatient clinic. You take a very small piece of tissue from the intestine, and from that biopsy you can isolate crypts. And the crypts of the intestine contain stem cells. And when stimulated with the appropriate culture medium, they start proliferating and forming into 3D structures we call organoids, or as we like to communicate to our patients, mini guts. And after a day or 7, you can disrupt them and use them for either passaging and keep them in culture, use them for different assays, or store them in a biobank. The assay we use in our experiment in this project is the forskolin-induced swelling assay, and also called the FIS assay. And forskolin is a Cftr stimulator. So what happens when you incubate or when you stimulate the organoids with forskolin, you stimulate Cftr. What we can see here is an example of organoids from a healthy control in comparison to organoids from donated from a person homozygous for F508del. And when you stimulate these 2 organoids with forskolin, you can see that after 60 minutes, the organoids from the healthy control really swell like a little balloon. And those of the CF patient don't show any response at all. And that's because the Cftr protein is on the inside of the organoid, like the lumen of the gut. And when stimulated, you get water secretion. The water fills in the lumen of the organoid and the organoid swells. We see a very large range of swelling from people with different mutations. And if you're interested in the correlation between the amount of swelling and disease progression, I would highly advise you to take a look at my colleague's poster number 665, but it falls a bit outside the scope of this presentation. Here we're interested in how we can rescue or restore CFTR function with drugs. And this is one of the earliest examples from Dekkers et al. Published in Nature. And what we see here is that when you incubate the organoids with, in this case, lumocaftor, ivacaftor, and then stimulate them with forskolin, that you can see swelling. And if you would inhibit it with a CFTR inhibitor, you don't see the swelling. This shows that the forskolin-induced swelling assay is a CFTR-dependent assay. And of course, the next question is, how does this swelling of the organoids correlate to response in the clinic? This is from a different paper where organoids were tested with different drugs derived from patients with different genotypes and correlated to the clinical response to this drug. And on the upper graph, you can see the correlation with, on the x-axis, the organoid swelling in correlation to the absolute change in FEV1. And in the lower graph, you can see the same correlation, but then with sweat chloride concentrations. And you can see that they, well, correlate quite well, and the different colors indicate the different genotypes. So during the first part of the project, called the HitCF organoid study, we were able to recruit 500 patients and obtain over 500 rectal biopsies from European and Israeli patients, all with ultra to ultra rare mutations, and performed 2 screens. Today, I will talk to you about the screen performed with ELX02, and last year we showed you the first results of the other screen. And the screen I'm going to talk about today is with ELX02 and was performed on 220 organoids. The criteria for these organoids to be screened with ELX02 was that they must have a minimum of 1 nonsense mutation, and of the complete group, 75% of the patient-derived organoid had 1 nonsense mutation and 1 other mutation, and 25% had 2 nonsense mutations. And what can be appreciated is that only 25% had 1 G542X mutation, which is the population where ELX02 currently is clinically being tested in. And on the map on the left side, you can see the distribution of the where the organoids came from. And you can see that the most organoids came from Israel and Italy. And this is quite in line with the geographic distribution of nonsense mutations throughout Europe. A bit about the working mechanism. So ELX02 is a eukaryotic ribosomal selective glycoside and knows its origin from aminoglycoside antibiotics, which are known to have read-through capacity. Here on the left side, you can see the normal situation. So you have an mRNA string, which is translated into protein by the ribosome. But as soon as there's a premature stop codon, which can occur at the beginning of the gene or towards the end of the gene, then at that point, translation stops and there's only a truncated protein developed, which mostly will be degraded by cellular mechanisms. ELX02 can bind to the ribosome and promotes premature stop codon read-through so that the full-length protein can be produced. One of the most important safety experiments done in their phase 1 trial is that they show that there's no evidence of native stop codon read-through, which is of course very important. Here we see the Western blot of 2 housekeeping genes, Cis-C and beta-2 microglobulin, of 2 patients from the phase 1 trial. And we can see that after 7 days of treatment with ELX02, only the native form of protein is shown and no read-through products can be found. And then an example of organoid swelling of organoids derived from a G542X homozygous donor. Here we see the swelling after adding forskolin when organoids are incubated in water, which is the negative control we use in our experiments. And this is after 48 hours of incubation with ELX02 and then stimulated with forskolin. You can see rescue of CFTR function, because like in the earlier example, CFTR is on the inside of the organoids. And when present and active and stimulated, it it enhances water secretion towards the lumen, and the lumen starts to swell, which can be microscopically be seen. And then to the most important slide of this presentation, and that's the responses of the organite from our organite screen. Here on the x-axis, you can see all the individual swellings. On the y-axis, you can see the area under the curve, so the relative swelling towards the start of the forskolin incubation. All these organites were incubated for 48 hours with 18 micromolars of ELX02, and then stimulated for 2 hours with 0.8 micromolars of forskolin. The responses that are shown here are corrected responses, meaning that if there was any residual CFTR function, so any swelling seen without drug incubation, that was restricted from the total swelling. The dotted line here represents the average swelling of homozygous G542X donor, which was taken along on every experimental plate as a positive control. And what you can appreciate here is that from all these patients with ultra-rare mutations, almost up to 75% shows to some extent response to ELX02, and even 25% shows a higher response than our positive control. So what we conclude from this is that up to 75% of patient-derived organoids with ultra-rare premature stop codon show swelling to ELX02, which makes ELX02 a very potent compound, which is currently in phase 2 trial for ultra-rare nonsense mutations, complementary to the G542X, in which currently clinical trials are being performed. And in the future, we would like to select the highest responders after a validation screen for a phase 2b/3 trial, which will be organized by Elox Pharmaceuticals and in collaboration with the HIT-CF consortium, and expected to start towards the end of 2022. Of course, we're very curious to deblind this, the genotype, so we can really see which genotypes respond and which don't, to assess the genotype-teratype relationship. And we would like to contact these patients and store their organoids in a biobank so they can be used for future screens with new compounds, changed compounds, or new techniques. So to get back to the main goal of the project, to get drugs available for all patients with ultra-rare mutations. A lot of people worked on this screen, on this project, but I would like to give a special thank to the people who did all the pipetting work, who had all the hundreds of organoids through their hands. Annelot, Sylvia, Annabella, and Iris, thank you very much. Of course, the entire consortium and the organoid study group, and not to forget all the patients who contributed to this project. Thank you all for listening, and I'm very happy to take questions.
Thank you so much for that excellent talk. So we have some questions coming in. The first one is from Tyler Brown, who asks, was the intestinal organoid model used because rectal biopsies are easier or less painful than airway biopsies? And have you tried an airway model?
Thank you, Tyler, for your question. The intestinal model was used because at the time the project was designed, we were way more experienced with intestinal organoid and the intestinal organoid model than the airway model. And indeed, I think the airway model is catching up on the intestinal model, and is indeed, especially the nasal brushes are easier to obtain than rectal biopsies. And currently we're going to do co-paired experiments in our lab to see how the airway model relates to the intestinal model. And I really think that, yeah, it might be catched up by the by the airway model indeed. But for now, we have more experience on the intestinal model.
That's great. Yeah, it's been very well established, and it's been exciting to see it mature over the years for sure. So we have another question from Carlos Perez Garcia. How is the variability in the response in organoids that have the same mutational phenotype?
That's a very interesting question. Thank you. So in this particular screen, we haven't deblinded the mutations yet, so I can't tell you answer this from this screen, but we of course did a lot of validation experiments within our own lab and between different labs. And what we see is that there is a high, it's a very low variability, but of course it's still a biological assay, so there is some variability. But being a responder or showing swelling to drugs never varies, in that sense that we, in the F5OADL lines to the currently available modulators, we always see a response. Not always to the same extent, but we always see swelling.
Okay. Another question. How does swelling in rare variant organoids compare to swelling observed in wild type organoids, for example, % wild type?
That's a very nice question. Thank you. I must say I don't know it by heart. So it's I think it's fair to say that I know it better for the shorter period of time, because if you swell wild type organoids for 48 hours, like we do here in this ELX screen, I'm not really sure to what extent they will swell, and I think they will reach their platform very, very quickly. But I have to look into that.
Okay, great.
Thank you.
Here's another question from Tanya Ganska. Why do you use water and not DMSO as a vehicle control for your experiments?
Because ELX02 doesn't dissolve in DMSO, but in water.
I know that for some of our other modulator compounds, they're not water soluble. And so I believe that, if I remember correctly, DMSO is used in those cases for intestinal organoids. And I just want to tag on a little question before I get to the next audience question. Do you notice a difference in swelling in the vehicle control using water and DMSO? Does that seem to impact your results?
Sorry, can you repeat that one more time?
Do you notice a difference when using water or DMSO as a vehicle control? Does DMSO have an impact itself on swelling?
No, it doesn't. No, thank you.
Okay, sorry. All right, now to the next audience question. Yana Lee says, very exciting data. How does the G542X organoid swelling with ELX02 compare to F5OADL organoid swelling with Trikafta?
I think what I would like to do to answer this question is to point out a recently published paper by a group where the swelling of where G542X organoids are screened with both ELX02 and ELX02 in combination with Trikafta and Nmd inhibition, but alone. So Trikafta on F5OADL is way more potent than ELX02 alone on G542X. So ELX02 needs add-on to come into the same range as Trikafta.
Here's another question from the audience from Zachary Sellers. Nice data. It will be very interesting to see how different genotypes respond to ELX02. As you may be aware, nonsense mutations are rare in white CF patients, but are more frequent in minority races with CF. It's more of a comment.
Yeah, it's more of a comment. Yeah, and I think so. What we're really trying to establish here within the HCF program is to lay the basis of precision therapy within this rare mutation using the organoid model. And I think as soon as we're starting off with the clinical trials, and when we're able to connect the clinical data to the in vitro data, and it works as we think it will work, then we can really start off with screening more patients, screening minority races, and be as inclusive as possible. Yeah.
I have a question for you. So right now, this is just a really exciting application of an in vitro assay to help, with clinical trial design in these, really rare mutations that would otherwise not be possible. But how might your results impact people outside of these studies or outside of Europe? For example, can individual mutation data be submitted to regulatory authorities to potentially expand access to other individuals with the same mutations? And what steps have you taken to get the kind of approvals that would be necessary for that to happen?
I think that is the ultimate goal, to have make the outcomes of this project beneficial for everyone who can benefit from it. I think the the steps towards the authorities are a bit too early. So I think we still need to prove the concept, prove that the clinical data is in line with our in vitro data, and then go on to the authorities. And so most of the questions we get regarding to the results is please deblind the data, and that's something that we're also very eager to do. But since we're still the pharma companies that are aligned to this project are still changing, and we're, well, hopefully can tell you some exciting news in the future that a new company is going to join us as well. So in order to be as well precise as possible, we still want to keep everything blinded, because if we now deblind a part of the organized and tell already to authorities you might be responsive, then we we're afraid that we're stop them from being able to test new drugs or different drugs. So at the end of the project, everything will be fully disclosed, and everyone who can benefit is able to benefit from these data.
Well, thank you so much for such an excellent talk. And I think we're just going to wrap up now. We only have a few more seconds. So thank you so much, and great job.
Good morning. This is Shamili from the University of Iowa. Today, I'm going to share our study in lunar efficient delivery mRNA in finite airway epithelial cells, in vitro and in vivo. This study was incorporated with the Cftr Therapeutic. Therefore, I have a disclosure with the Cftr as a concerning research concerning. Despite the development of Cftr modulator in CF disease treatment, gene therapy for CF disease remains an alternative approach for CF patients, those who have no response or less response to Cftr modulator therapies. In addition to the viral vector-mediated gene transfer, no viral mediated gene transfer, particularly the recent mRNA medicines, have demonstrated great potential in treatment of many diseases and the vaccine development. For example, the COVID-19 vaccine we currently use. Therefore, the Cftr Therapeutic developed a novel lunar lipid for mRNA delivery. This offered the opportunity for testing the mRNA therapy in CF lung disease. To do that, we need a great animal model that are able to recapitulate CF lung disease in humans, because the current mouse CF model have no lung disease phenotype. Therefore, Dr. Inhart at the University of Iowa created CF ferret models. These CF models could develop both of the lung disease phenotype and the pancreatic disease phenotype, as seen in the CF humans. In addition, they also create a transgenic LOXP report transgenic ferrets. In this ferret, the membrane-bounded tomato and the membrane-bounded EGFP on and off transgenic assay were inserted in the local locus of the genomic DNA of the ferret. In the presence of the recombinase, the tomato transgene gene can be converted into EGFP fluorescent proteins. By using the ferret models, we tested lunar human CFTR mRNA, lunar Korea mRNA, and lunar TD tomato mRNA in the ferret airway epithelial cell in vitro and in vivo. The lunar Korea mRNA delivered to the LOSA transgenic ferret of the polarized airway epithelial or in vivo lung will result in the conversion of the EGFP And the EGFP converted cells can be used as index for gene transfer efficiency. Similar, the LONA TT tomato mRNA transduced in the wild-type fatty long or porous airway epithelial cells, the transgene of TT tomato mRNA will also can be detected by the fluorescent TT tomato fluorescent proteins. To test the function of the LONA human CFTR mRNA, airway epithelium generated from the epithelial cells from the CF fillet, and the transduction of the mRNA will result in the CFTR functional correction that can be assessed by the measurement of the CFTR-mediated chloride ion transport using an in-using Trimble system. First, we tested the LONA lipid-mediated mRNA delivered in the fed airway epithelium in vivo by using the LOSA transgenic fillet airway epithelial in vivo model. The transfection of the LONA mRNA and will result the conversion of EGFP in the epithelial cells at 48 hours following the transfection. Similarly, the wild-type fillet airway epithelium generated in vivo is well differentiated with abundant cellular cells, and the transfection of the LONA TT tomato mRNA caused the tomato protein expression in the epithelial cells at 48 hours after transfection. In order to test the functional restoration of the CFTR, the G501T cell-fed airway epitheliums were cultured at the air liquid interface for 3 weeks, followed by the transfection of the LONA human CFTR mRNA from the apical side of the epithelium. 48 hours later of the transfection, the CFTR function can be detected in the using a using chamber to assist the CFTR-mediated chloride ion transport. The recording of the ion transport demonstrates the transfection of airway epithelium cells transfect with LONA's human CFTR mRNA, significantly increase the CFTR-mediated chloride response. And this response is similar to that in by the epithelium treated with the VX717 CF modulator for G551D as compared to the control epithelial cells. Quantification also demonstrates a dose-dependent CFTR functional correction. The high dose of the LONA human CFTR mRNA transfection lead a highest of the CFTR function correction. However, the dose, the 25 microgram per ml of transfection in the failed airway epithelium see no significant changing of the CFTR function correction. The overall correction is like in transfection of the CFTR mRNA at a dose of 100 microgram per ml, lead about 6-fold high CFTR-mediated chloride ion transport compared to the LONA DT tomato transfected epithelial cells. However, of note, we need to mention the high dose of the LONA formulation infection also caused the reduce of transepithelial resistance in the failed airway epithelial cell in vitro. In agreement with CF functional studies, a dose-dependent increase of CFTR protein also was observed in the LONA CFTR mRNA transfected epithelial cells, as determined by the immunoblotting assays. Next, we tested the efficiency of the LONA lipid-mediated mRNA transfection in the animal model in vivo. So the LOSA transgenic fillet was intratracheally delivered with the non-nuclear mRNA in the lung. 10 days later, the lung was collected for analysis the GFP as the transfection efficiency. The in-phase images show abundant EGFP particle cells in the surface of the trachea epithelials, similar in the intralobar airway epithelial cells. In order to identify which epithelial cell type was transfected by the low-nuclear mRNA, immunostaining with the epithelial cell marker alpha tubulin was performed. As seen in the images, the majority of the GFP particle cells also express the cellular cells, demonstrated that this cell type is the main cell type for low-nuclear transfection. In addition to the cellular cells, with the goblet cell marker staining MAC5A system, we also see the co-localization of the EGFP and the mucin 5AC, suggest MAC5AC and the other secretaries also can be transduced by the low-nuclear mRNA. The quantification study demonstrates about 36% the overall of the trachea epithelium cells can be transduced by the low-nuclear mRNA. Among them, 16% is cellular cells, and about 12% is the secretory cells. In the intralobar airway epithelium cells, as high as 40% of the total epithelium cells can be transduced by the low-nuclear mRNA. Among them, 24% is cellular cells, and 40% is secretory cell type. To test the efficiency of LONA TT tomato mRNA transduction, the Y type of fate was intratracheally transfected with LONA TT tomato mRNA, and the transgene expression was detected at 48 hours after the LONA delivery. As seen in the in-phase image, the abundant TT tomato particles was observed in the surface of the trachea epithelium cells. As seen in the low-nuclear delivered fillet, the majority of TT tomato particle cells was also cellular cells, same as seen in the bronchioles and the intralobar airway cells. Quantification study also demonstrated about 24% of total trachea epithelium cells was transduced by the LONA TT tomato mRNA. Among them, 60% is the cellular cells, and 4% is secretory cells in the trachea. In the intralobar airway, we also see about 36% of total epithelial cells was transduced by the low-nuclear tomato mRNA. Among them, 8% is cellular cells and 7% is secretory cells. Of interest, we also observed abundance for the alveolar cells was also transduced by the LONA formulation made at mRNA transfection, either in the low-nuclear mRNA transfection or the LONA TT tomato mRNA transfection to identify which kind of cell type was transduced in the alveolar region was transduced by the LONA reagent. Significant protein C at alveolar type 2 cell marker was co-standard within these tissues, and the result demonstrate about 40% of the alveolar cells can be transduced by the low-nuclear mRNA. About 24% of the alveolar cells can be transposed by the LONA TT tomato mRNA. However, about 60% of the alveolar type 2 cells was transduced by the LONA reagent made mRNA transfections. In summary here, fillet are useful model for studying the pathology of an organ affected by a CFTR mutation, including the lung. The low-size transgenically reported fillet are an excellent tool for testing gene transfer vector in vitro and in vivo. LONA formulation efficiently transfect to various epithelial cell types infiltrate airway in vitro and in vivo. Low humans Cftr mRNA delivery restored Cftr mediated epithelial chloride transport in CF-filled airway epithelial cells in vitro. Further studies in CF-filled model using lunar human Cftr mRNA will demonstrate the potential of mRNA therapy in CF lung disease. Lastly, I would like to thank Dr. and his member in his lab for their assistance in this project, and Dr. Carlos Casillas group for their support in collaborate in these studies. With that, I would like to stop here and take questions. Thank you for your attention.
Thank you, Dr. Liu, for that. I think we're just waiting to get the, there you go. Hi. Thank you so much for the wonderful talk. I have some questions for you coming in from our audience. Rihanna Lee says, very nice data. Was there any immune response to the lunar system in your in vivo model?
Honestly, we didn't detect any perform this kind of testing. Currently, we don't know.
Okay.
But it's a good idea. We will plan to do it in the next step. This is a great question, and it's truly a concern for gene therapy.
Great. I have another question from Cal Perek. Can you transduce glandular cells in the airway? Have you seen evidence of that as well?
Yes. But only we can see some in the glandular dark region when we use both of the lunar chloride mRNA and lunar tomato mRNA. Yes, we do see, we did see the transfection in the submucous gland.
Okay. And I have a question for you. So you showed data that you had uptake of the reporter all the way into alveolar cells, as well as into the intralobar bronchus. And I just wondered about the differences between the ferret lung and the human lung. Would you expect that we would have that distal spread of the construct in human lungs as well? Do you anticipate that it would reach the small airways or even the alveoli for humans?
We don't have any test data, but I expect it will be because from the anatomically and I think physiologically, the ferret lungs very similar to the humans, and because technically we use the a gene delivery through the intralobar airway. They will go to the fairly distally when we see a couple of the gene transduction strategy on the other vectors, including the viral vector and the no-viral vector. We do see in the ferret lung, with the gene transduction. So we don't have any human data or human gene transfer, but I expect it might be works for the delivery in the distal lung.
I mean, just a quick follow-up for that before we get on to the next question. I believe you said that you use an atomizer to do intratracheal.
Yes.
Is that more similar to like an aerosol or a nebulized therapy in humans? I'm just not sure how that relates to what we would have available to us for airway delivery.
So thank you for the great questions. Actually, we all know this is clinically that strategy might be not applicable for gene transfer. You need the nebulizer. So, however, the nebulizer need the efficiency. I mean, for failure might be take a 4 ml of the reagent might be 90% would be in the load because during the delivery cannot be reaching the lung. So So for this study, we try just like for the concept proof for the gene correction or gene transfer. For the next step, we're moving forward to the neprilysin for delivery this in this models. That's great question.
All right, so another question from Calvin Cotton. Was in vitro ALI lunar delivery only to the apical compartment?
Yes, we did it from the apical compartment. But we also use like in basolateral compartment, it also works because when you add the transfection efficient reagent in the basolateral side, they also can be like protected by the epithelial cells as well. But for this study, we did it in the apical side.
I have another question for you. So this is a lipid-based formula, obviously. What barriers do you think exist to trialing that and translating it to humans? Like, have you, earlier our audience member commented on, immune response, but are there any other adverse responses that you've seen within the ferret lung? You mentioned decreased area epithelial resistance, which is at the highest concentration, which suggests some damage, but any other barriers that you have considered with using this in the lung?
Yes, I think the most tough, is because in the CF lung, the thickness of the mucus, the penetration of the mucus is more it's challenging. So we do try to use if we did wash off in the in vitro study, we wash off the mucus in the apical side before we added the transfection the reagent. So in vivo we cannot do that. So next step, we're trying to use the CF ferret with a deformed lung disease and try to see if this reagent can be efficiently penetrate the mucus layer in the ferret epithelial airway. I think the tough challenge is the mucus, how to penetration the mucus layer.
We have time for a couple more questions from the audience. We just have one more minute left. All right, well, I'd like to thank you so much for your great talk. It's so interesting, and we're looking forward to seeing how this progresses as you expand to other, to your CF models and do those additional work. It's very exciting.
Thank you, Jennifer.
Hello, my name is Craig Hodges from Case Western Reserve University, and I would first like to thank the moderators for the invitation to present in this workshop session. My talk will focus on poster 662, describing our panel of mouse models in which we've replaced specific mouse exons of CFTR with homologous human exons of CFTR. The goal of this study is to provide the research community with in vivo models with human CFTR sequence to test potential gene editing therapies. I have no relationships to disclose related to this presentation. So why create human CFTR exon replacement mouse models? I will answer this by splitting this question into two questions. First, why the mouse? Mice are easy to genetically alter, are a model with low costs, quick to expand, as well as CF mouse models have led to various insights into disease, have proved useful in studying CF, and they display aspects of most CF disease manifestations. Second question is why create exon replacement models? With the explosion of new technology in the gene editing field, we need human sequence in an in vivo model to test delivery and efficacy of new potential gene editing therapies. There is 82% sequence identity in the coding region between mouse and human CFTR. An example is shown here comparing human and mouse exon 12. The human sequence at the top, the mouse sequence is at the bottom. As you can see, the green to yellow nucleotide changes are synonymous base changes that do not lead to an amino acid change. However, the red to black nucleotide changes are non-synonymous base changes that do lead to amino acid differences. These 11 changes or 11 differences would complicate things when trying to establish gene editing therapies. And even though there are differences between human and mouse CFTR with respect to folding, gating properties, several studies have shown that chimeric human-mouse CFTR proteins are functional. Here at Case, we have made other humanized mice with the full 188 kb human CFTR gene, along with surrounding regulatory regions. We have also created specific human CFTR mutations in this model. These models are proving very useful for testing CFTR modulators in some gene editing strategies. However, the way this mouse was originally created, in which the large CFTR transgene randomly inserted into the mouse genome, multiple tandem copies inserted, which can make gene editing strategies difficult and results hard to interpret. To this end, exon replacement mice will hopefully prove to be an improved gene editing tool for the CF research community. Now that I have hopefully convinced you of the need to create these exon replacement mice, let me show you our strategy. Shown here is our design for replacing mouse exon 12, shown in the gray box, with surrounding introns shown by the gray lines. We designed guide RNAs, shown as these arrowheads, that targeted about 200 base pairs on either side of the exon. We injected these guides along with Cas9 and a plasmid containing the desired sequence to replace mouse exon 12. Shown here is wild type, or normal human exon 12, shown as a white box that replaced mouse exon 12 in the endogenous mouse CFTR gene. The replacement includes the whole exon and about 150 to 200 base pairs of human introns around the exon, shown here in black lines. To preserve sequence for correct splicing. This replacement would lead to normal nucleotide sequence as well as amino acid sequence for just the human exon 12, while the rest of the gene would be mouse CFTR. Even though previously published studies suggested chimeric CFTR proteins should be functional, we created the wild type version to verify that it complements loss of mouse exon 12 and is fully functional in the replacement. This strain will be referred to as H exon 12 in my slides to the rest of the presentation. In addition, we created a strain that contained G542X in the human exon 12 using the same design. We chose to start with specific nonsense mutations for these exon replacement mice, since there are no FDA-approved modulators directed towards such mutations in CF patients. After injection of the necessary components into one-cell mouse embryos and transfer into pseudo-pregnant females that gave birth to resulting mice, we identified founders and eventually bred heterozygous mice with these exon replacements to make homozygous mice. Two strains, human exon 12 and G542X human exon 12, were made. We then examined CFTR expression, function, and CF manifestations in these two strains compared to wild-type mice and CF mice with G542X in mouse CFTR that was previously made. We first looked at CFTR expression in several tissues from homozygous mice from these strains and compared mice with wild-type CFTR mouse sequence as indicated by this line here at 100%. The human exon 12 mice in dark gray showed similar levels of CFTR. However, the human exon 12 with G542X, shown here in white, showed significantly reduced levels of expression due to nonsense mediated decay, similar to mouse G542X, shown here in the light gray bar. We then wanted to assess CFTR function in the airway by assessing nasal potential difference. The nasal potential difference across the nasal epithelium was measured before and after the addition of a chloride-free solution containing forskolin. Forskolin stimulates adenylate cyclase, leading to activation of CFTR through increased cyclic AMP levels in protein kinase A-mediated phosphorylation. Non-CF animals display chloride secretion in response to this challenge, generating a lumen-negative potential difference, shown here in wild type and human exon 12. While CF mice do not respond, as shown here in human exon 12 with G542X and mouse G542X. We looked at CFTR function in the intestine using intestinal sections from mice in an Using chamber and observed the short circuit current before and after CFTR activation. We see wild type and human exon 12 mice showing robust CFTR function, while both mouse and human versions of G542X are significantly reduced in each of these tissues. Surprisingly, in the nasal PD and the jejunum, the human exon 12 wild type compared to wild type mice are actually increased for CFTR function, and we're investigating this. A prominent CF manifestation in both humans and mice is reduced growth. CF mice display a clear reduction in growth throughout their lives, as shown here in 4-week-old mice. Here are weights from 6 to 7-week-old mice from these 4 strains with an equal number of males and females in each group. The human exon 12 mice are identical to the wild type mice, while the G542X is reduced significantly, similar to the mouse with G542X. Another prominent phenotype in CF is intestinal obstruction. In CF patients, this presents as meconium ileus at birth or distal intestinal obstruction syndrome and constipation later in life. In CF mice, intestinal obstruction presents around the time they are starting to eat solid food, and without a laxative, this can result in death. Human exon 12 mice are similar in wild type mice, showing no survival issues. As shown here, the gray and the black line are on top of each other. However, CF mice show intestinal obstruction and quite a bit of death right here around when they're weaned. Both the human exon 12 G542X as well as the mouse G542X shows this. The next thing we did was test whether human G542X could be corrected. We utilized an in vitro assay using primary intestinal cells called intestinal organoids. These intestinal organoids contain crypt-like structures and an intestinal lumen lined by differentiated cells, recapitulating the in vivo tissue architecture. Intestinal CFTR is predominantly expressed at the apical membrane of the crypt cells, where its activation, in this instance using forskolin, drives the secretion of electrolytes and fluids, leading to the increase in size. The use of these organoids to test CFTR function was really pioneered by Jeff Beekman's group in the Netherlands. Here's an example of wild-type mouse intestinal organoids with forskolin added. You can see almost all organoids swell in a short period of time. We measure how much all the organoids in a field of view expand as a percentage area increase. If these organoids do not have any functional CFTR, they do not swell. Here's a forskolin added with human exon 12 wild type version, and this swells very quickly, similar to the wild type of the mouse. However, the human exon 12 with G542X over the same time period shows no swelling, indicating that there is no CFTR function. Here, we added G418, a read-through agent, as well as a nonsense mediated decay inhibitor, SMIG1I, to the G542X organoids, and showed within an hour there was swelling, but it was quite delayed and not nearly as robust. When we added correctors to help CFTR folding along with the read-through and nonsense mediated decay inhibitor, we saw increased swelling and robust CFTR activity. With the area the organoids measures as an increase, percent increase of the starting area and a minimum of 4 wells per group, you can see here that the human exon 12 is identical to wild type organoids, indicating that really there's no deleterious effect of replacing mouse exon 12 with human exon 12. Human exon 12 with G542X, swelling is shown here with forskolin alone, G418 and SMIG1I, as well as all of the modulators, the correctors, G418 and SMIG1I, showing the most correction. This can be compared using area under the curve shown here, where wild type and human exon 12 are identical, and increasing function of G542X restoration is shown here. However, they are all significantly different than wild type. However, really, we created this model to see if we could establish gene editing. So next, we utilize these organoids to test if we could do just that. We accomplish this by taking human G542X organoids and digesting them into single cells, adding a guide RNA that recognized G542X mutation, along with Cas9 nuclease and human exon 12 correcting oligo. We electroporated these, and only single stem cells that are still alive will grow up into individual organoids. These can be stimulated with forskolin to test for CFTR function. The great part is that because these started from single cells, each organoid derived from a single stem cell may or may not have been gene edited. So each organoid is a single event of potential gene editing. Here's a representative well of organoids that are gene edited. Remember, each organoid is a single potential gene editing event. You can see that several of these organoids have swollen within the hour. We find that on average, 11% of these organoids swell versus 0 of these organoids just with Cas9 and only 1 or 2% without the corrector. When we sequence this population of organoids, we find that indeed wild type sequence does exist, but only in this group. We plan on taking these individually swelled organoids, subcloning the population, and then comparing sequence with a pure population of edited organoids to compare. Currently, we have created wild-type human exon 12, G542X human exon 12, as well as R553X human exon 12. Now, we just started characterizing this particular R553X mutation, so I don't have that data in this talk. In addition, we've created human exon 12, human exon 11 wild type version, as well as F508del for those of you interested in gene editing, the most common CFTR mutation. Then coming soon, we'll have mouse exon 23 replaced by human exon 23 with the wild type version, as well as W28X. As a side note, if you're interested in W28X models, check out poster 661, presented as a talk in workshop 12 by my graduate student, Margaret Michicic, who talks about other in vivo W28X models that we have created. In conclusion, we have successfully created wild-type and G542X human exon 12 replacement mice. We've shown that human exon 12, the wild-type human exon 12 strain has normal CFTR expression, function, and no CF manifestations. The G542X form has similar CFTR expression, function, and manifestations as mouse G542X. This mutation can be corrected in vitro through pharmacologic and gene editing means. And we're really looking forward to collaborating with other groups to see if we can gene edit in vivo throughout the whole body in these mice. These future exon replacement strains, F508del, R553X, and W28X, will also be available. I'd like to acknowledge all the people involved in the research presented here from CASE, as well as CF Foundation for funding. If you're interested in any of these mice, you can talk to me, Ron Conlon, or Mitch Drum, and you can also check out poster 662 for a summary of this talk. Thank you.
Thank you, Dr. Hodges, for such an excellent talk. Again, I invite the audience to put some questions into Slido. And while we're waiting for those to come in, I'd like to ask a question myself. You alluded to this at the end of your talk, that you were going to have these available for the community. Can you elaborate a little bit on how that will work as they become, as you continue to make more and more of these models?
Yeah, thanks, Jennifer. For those of you who aren't aware, here at Cftr Reserve University, we actually run a CFF-funded Mouse Model Resource Center. And so we've made quite a few of these mice in the past, and they are available to people. You can go on our website, cfmice.org, or contact me directly. With these, as long as we're confident with the, after we characterize them and are confident with the sequence as well as the function, they will be available. So.
Great. Thank you. That's going to be a great resource for sure. All right, so we have a couple questions coming in from the audience. So from Jae-Suk Young to Dr. Craig Hodges, the 3' UTR may affect stop codon, the NTC and or PTC termination efficiency and suppression. Human exon 27, which I guess the human exon 27 replacement might also be interesting. Can you comment on that?
Right now we don't have, I guess we don't have any plans to replace exon 27, but I agree that there's certainly differences there, and that would be interesting. We started with the exons with the most common mutations, as you can probably tell, to try to provide the tools that would most likely be used by the majority of the community, but we're certainly open to others' ideas and working with them to maybe create other exon replacements that might be needed.
We have a couple questions from Patrick Harrison. Nice work, Craig. One of your early slides showed human exon 12 was statistically better than mouse exon 12. Any idea why? And then how much human intron 11 or 12 was included either side of the human exon 12?
Yeah, so thanks for the question, Patrick. Yeah, we were kind of stunned by that too. We're not really sure on it was more on the nasal potential difference assay that both, we actually saw the same thing with human exon 11 replacement, even though I didn't show that data. The function of CFTR in the nasal epithelium was significantly stronger than in wild type mouse. So we're looking at that. I actually don't have any ideas at this point, but I do think it might be tissue specific, just because in the intestine, while I think there was some significant difference in the jejunum, the other tissues really didn't show any difference in CFTR function. I wish I had some ideas of why replacing a single exon with human might make it a little bit better. I'd have to compare the sequence to see if for some reason it's more codon optimized than the mouse, but that's probably the only idea I could come up with. And then as far as how much intron around the exon was replaced in each of the models, we tried to include about 200 base pairs of human intron on each side.
All right, just waiting for more questions from the audience, but I have another question for you. So I have an interest in organoids as well, and obviously we had a talk earlier about using human or intestinal organoids. What gave you the idea? What was the advantage of using the intestinal organoids taken from this mouse model to do additional testing?
Well, organoids in general, and you know, as the previous speaker had shown, are just so dynamic and can be used very quickly in showing the restoration of CFTR function. And so even though we do have human organoids available, sometimes getting those with the right combination can be a little bit difficult, of mutations can be a little bit difficult. So in this particular case, when we made these mice, we used the organoids 1. It's a nice way to combine quite a few different compounds very quickly to see what might work in vivo. As well as the gene editing part has been nice to show that kind of as a representation of what might occur in the whole animal or throughout the body if you could get the right delivery strategy. I know there's a lot of experts out there working on the right delivery strategy, and we're hoping that these models will help answer some of those questions of have they delivered enough of these gene editing components to restore CFTR function in vivo.
Yeah, that makes sense. Thank you so much. We have a couple of other questions come in. The first one is from Calvin Cotton. Do you know if human exon 12 confers sensitivity to ivacaftor to mouse CFTR?
So we actually know, just to give a little background on that. So we know wild type mouse, it does not respond to VX770 or ivacaftor. We haven't tried it on human exon 12. We have tried it on a couple other mutations and found, it didn't help at all. I believe that there was a publication that showed there was a part of CFTR that kind of conveyed this sensitivity to VX770 that was different between mouse and human, and it wasn't in exon 12. So my answer is it probably does not, but we can certainly try that. I believe that difference was somewhere else within CFTR.
Another question, this time from Martin Mintz. How conserved are the splice site acceptors and donors between human and mouse? And could those shift mRNA isoforms and possibly account for better CFTR function in the exon 12 replacement?
That's an interesting idea. As far as I know, I think that the actual splice site acceptors and donors are very conserved between mouse and human. So could the surrounding sequence kind of outside what we typically think of as the splice site and acceptor and donor be different? That's a possibility. So yeah, we'll have to look at that. Uh-oh. Oh, are you still there?
I am. I have a blip in my internet. So, hey, we got through the whole workshop with no technical problems so far. So I think we did a pretty good job this time. All right. Well, oh, here you go. Here's another question. I think this is also this is from CalPrec. So Craig, another great model from your core. Congratulations.
Sorry, this is the question from Carlos Perez Garcia. I just didn't have time to type it in before you.
Okay, thank you. Is this there's been some talk during the conference about super exons. Have you considered this approach in your mouse models?
Yeah, that's a great question. Actually, we're in the process of working with Hillary Valley from the CFF labs. Ron Conlon and myself have been in talks with Hillary and Cal Cotton there from the CF labs. And we are working on an approach to actually insert some of their super exons that they found work in vitro into our mouse models. So that certainly is on the way.
Great. So I think I'm being told that we need to wrap up after this question, but I think we have a few more minutes for sort of additional questions. I think that would be okay. So I'll have one more question in the chat. So from Patrick Harrison, he's asking for his earlier question, is splicing possibly the answer? And let me see if I can remind you what that question was about. About the human exon 12 being statistically better. Could splicing potentially be the answer for that?
Yeah, I think that kind of goes along with what Martin had asked as well, and I'm certainly open to that theory that it could be that there is something about the splicing that makes the human exon 12 somewhat better. That's certainly true. We'll have to look into that. So. All good ideas.
All right, let me just check. Any other questions for Dr. Hodges? Just checking to see if maybe we can open up in these last couple of minutes that we have to all speakers, because there was at least one or two questions that came in a little bit late, and since we're at the end of our Q&A. So I guess if you guys do have any questions, the contact information for all of us is available in the conference, please. Oh, well, now I'm saying, so hold on. Now I'm saying that, yes, I do have 6 minutes left. And so I would like to invite all of the speakers to turn on their cameras. And for the audience, if you have any additional other questions, we can answer those now. So the first question is for Dr. Oliver. It's from Martin Menz. It says, your data is very interesting. I was also wondering, what are the implications of this for therapies? Do we need to do theratyping of different modulators on specific SNPs? It's hard to imagine that we can fix those SNPs on their own.
Yeah, I don't envision there being, for example, small molecule therapies that would target silent polymorphisms. I just think it's an important factor to not discount when, for example, you may have a patient that, according to their disease-causing variants, should be responsive to a modulator, and perhaps they're not, or even, we might consider that particular mutations that might not even be on label for something like Trikafta, if they have a silent polymorphism like this T2562G SNP, that might influence the way in which their CFTR is folded, and therefore it might be a bit more correctable when you add something like Trikafta on. So I think in a perfect world where person power and funding are not limiting factors, I would say, yes, it'd be great if we could, do full, intron and exon sequencing of patients and identify whether they have silent polymorphisms like this and whether that may influence their pharmacologic response. But I guess that we're going to have to perhaps, work together with the care teams and the newborn screening clinics and everybody together to figure out the best way forward to make sure we're not missing people who might be responsive with modifiers such as a silent polymorphism.
I'd like to ask a follow-up question on this. Is there any reason why these silent polymorphisms might not show up in a GWAS, for example, or similar, sort of genome-wide assessment to find a modifier for modulator response or potentially variation in phenotype? Because it seems like, we might have seen it there, but on the other hand, it's considered a silent polymorphism. So I think it would, if it had been shown up, it quite possibly could have been discounted. So do you have any comments on that?
Yeah, I think you're right. It probably would have been discounted in a situation like that. There's also a part of me that wonders, when we get sequencing results back from a lab to identify patients' mutations, are we really taking a look to see very closely whether they have the most abundant codon, that's traditionally associated with the disease variant? So like in the D614G example, the GGC codon is traditionally what is reported as being, prevalent in CF patients. But what if that revertant that we engineered, what if that also occurs in patients and we don't take a deep enough dive down? We just take a look and see like, okay, well, this is the amino acid change, but we're not actually looking for the codon exchange itself. So I think, maybe it just might take a deeper assessment of those sequencing results to identify, okay, we've got patients who actually have different codons for particular disease-associated variants that could be important. But in addition, these silent polymorphisms. But then your question about GWAS is, the way that they're traditionally conducted, I really think that you would probably miss silent polymorphisms just for the reason you mentioned earlier. But I also have to think back to our example here where, when we were surveying the CF population, that the G nucleotide was not very prevalent among the entire CF population. It was only when we assessed those alleles that did not contain the delta F that we found that it was increased in abundance, very, very much enriched for prevalence there. So it would probably take stratification of GWAS data to see polymorphisms like this emerging as of interest.
Very interesting. All right, are there any other questions from any of our workshop speakers or anyone in the audience? I think this was a great session. Thank you all so much for your great talks and your great science. And I think, we are continuing to discover all sorts of new things that are going to be applied not only in CF, but in other diseases as well. And we're going to figure it out, I think, and that's exciting to know. So, all right, well, thank you guys all and enjoy your conference and have a great day.